Hello students, a warm welcome to all of you in our 7 days online certification course on topic NGS. Okay, in today's session we discuss about the differential gene expression. In today's session we discuss about the differential gene expression. Okay, so we start today's session and first we understand what is a differential gene expression. Differential gene expression in next generation sequencing is especially for the RNA sequencing experiment. Okay, with the help of this sequencing, we easily compare the two sequences, two genes at their expression level. Okay, with the help of differential gene expression, we compare the two genes at their gene level in different conditions. Different conditions like one gene is one condition of one gene in treated position or untreated position, in diseased person or healthy person, in mutated cell or in normal cell. Okay, we check the comparison between the two sequences or two genes at their expression level or gene level. Okay, now what is the basic concept? Basic concept is every cell expresses a subset of its gene and that gene as a RNA, because after the transcription process this DNA converted into the RNA, then this RNA converted into the protein, and protein is a functional unit. Okay, and this mRNA contains some genetic codon and basically triplet form of nucleotide is known as a genetic codon. It is coded by by the help of ribosome on the ribosomal and DNA and protein are synthesized. Okay, this is a different perspective. Okay, but why okay, this is using, we can sequence all the RNA molecule. First, we isolate the mRNA, then we convert this mRNA into the cDNA. cDNA is a complementary DNA. Then we perform the sequencing. Okay, by comparing the number of sequence read to map each gene across the sample, we can determine which gene are expressed more or less in different conditions. This is the main prospect of basic concept of differential gene expression. In differential gene expression, we target the RNA. First, we isolate the RNA, then we convert this RNA into the cDNA, then we perform the sequencing, then NGS. Then we perform the NGS or sequencing portion, then we analyze this result by the help of bioinformatic tools. Okay, by the help of this differential gene expression, we find out the comparison between the two genes in different conditions, like a condition is treated or untreated, disease or healthy, or mutated or normal. Okay, now this is the complete workflow of differential gene expression. First is your isolation, then NGS, then sequencing. After the sequencing, we check the quality. For checking the quality, we use the FasterQC and MultiQC software. Then trimming. For trimming, we use this Trigonometric and Adapt. Okay, and also we use FastP. Okay, then alignment. For the alignment, we use HischeckTo. For reading count, for generate the read count, we use feature count or head sequence count. Then for the normalization and differential gene expression analysis, we use this D is equals to HR and limit. Okay, these all tasks are performed on Ubuntu, and this is performed on RStudio. Okay, then functional annotation or visualization. For this, gene anatology, KH pathway analysis. Okay, KH is a pathway and gene anatology is all. For gene anatology, a database is present that is known as a David. For KH is a database. This is the database for the pathway analysis. Okay, this is the complete workflow of differential gene expression. Now, first step to quality check. What is the quality check? How we check the quality? We already discussed in detail. Okay, and this is the command name. Your FASTA QC, then your sample, then output, and then folder name in which you want to download this result. Okay. Next is your trimming part. Trimming if required. If does not require, then skip this part. Okay. Next is your alignment. For the alignment, we use Step 2 software. For Step 2, here we require the reference sequences. So we download the reference sequences from different databases like NCBI. Okay. After this, and we also install this hashtag software. After installing this, we run this command, Hiscat2, then -x, then your genome index, then your reference sequences, then SAM. Okay. After the alignment, we find the SAM file. Then we convert this SAM file into the BAM file by using this command. Then convert this BAM file into the short file by using this command. Then perform indexing by using this command. Okay. First alignment, then SAM, then BAM, then short, then index. This is the complete workflow of alignment. After this, maybe we find this SAM file, BAM file, sorted file, and sorted.bam file. Okay. Now the result of this alignment is used for the feature count. For the feature count. What is the feature count? We already discussed in detail. Okay. First we install this, then we run this command. Here we also require the annotation file in GTF format. Okay. For downloading this file, we use the database like Decode or NCBI. Okay. And this is the command. Okay. Output your gene, column sample, and values like a raw data counts. Okay. Then all these tasks we perform on Ubuntu or WSL. Then next step is your differential gene expression. For the differential gene expression, now we move to the RStudio. Okay. Here first we download the package. We download the, if we want to use this D sequencer, then we download this. Okay. First step is installation. We install this, and there are other software also available. We also install that particular software. I will discuss this. Okay. Then we load the library by using this command. Then input data by using this command. Count data. Then input the data. For inputting the data, we use this command. Okay. And counted text, this file, or we find by the help of this file we get after the feature count. Okay. Then we give the conditions. Okay. After running this command, then after preparing the sample means metadata. This is the now we prepare the input data. For inputting data, we require the file of feature count. Okay. Then we create a metadata file like condition. Here control and treatment means different environment. Count data, read dot table, then count text, header true, row name 1. All data, read CBC file, metadata, CBC, row name 2. And this is the command. Okay. After preparing this metadata, we run the DEG. Okay. Then filter out the significant gene if by using this command. Then finally, we save the data. After saving the data, we find MA graph, volcanograph, heat map, and PCA plot. And MA plot is used, the purpose of MA graph, log to fold. This MA plot, the purpose of MA plot, log to fold changes versus mean expression. Volcano plot for the significant up and down gene. Heat map expression pattern across the sample. PCA plot for the sampling clustering. For this, we use this package or this software. We also install all these software in our R Studio. Okay. Okay. Now, now we discuss. Now we discuss the complete process of differential gene expression. Okay. For differential gene expression, first we activate the RNA environment by using this site, that is the conda activate RNA sequence. Then we move to this D folder like CD, MENT, D, then LS. Now in my drive, these are the folders present. Now I move to the my required format, that is the DNA barcode. Okay, in this DNA barcode, these all are the folders are present. Now my required folder is a human gene. Now in human gene, now I check how many files are present in my gene. In human gene, these are the files are present. Okay. Then first we check the quality. For quality, we simply run FastAccuracy. Okay. Then we perform the alignment process. For alignment process, first we install this Histec2 software. For installing this Histec2 software, Histec2 software, I using this command. Okay. Then this Histec2 software is installed in my PC. Okay. Now then after installation, I run this command. That is the Histec2 genome, Histec2 space -x hg genome, then space, then -u, then your my sample, then SAM. Okay. Then we find this alignment result. Okay. Then by the using this command, I validate this result. Means these are the top 10 result. Okay. Then we convert this SAM file into the BAM file. Okay. Then validate this result. Then convert this BAM file into the short file. Then validate. Then this short file is converted into the indexing. Okay. Now to validate this complete result, I use this command, that is the SAM view. First I use this command SAM to quick check SAM short.bam and sort.bam. Now for checking the complete tool, I use this command. My BAM file is okay or no. Okay, now my BAM file is okay. Okay, then we use this command to validate our result, only to see the top 20 result, and these are the top 20 result. Chromosome number, their sequence, starting point, end point, and their sequences. Now the alignment is completed. Now we move to the now next step is feature count. For feature count, first we install this feature count software. For installing the feature count, I use this command, that is the app install subfield. Okay, then this feature count is installed in my PC. Then we check the version. Then we run this command, feature count -t ref sequences. Then we find this result. Then we validate this top. Then we easily visualize the top 10 result by using this command -head space -n 100 counters. Now these are the result. Okay. Now we move to the another part, that is the differential gene expression. For differential gene expression, I use RStudio. First, we check the package in my system. All these packages are installed. Okay. Then we define the path by using this command. Okay. Then in my system, a package is not installed. That is this package. Now I install this package. For installing this package, we use this command. Okay, now this package is installed. Now we check means this package is installed or not. For checking this, we use this version. Okay, now I load the library. For loading the library, now we import the feature count file in this RStudio. For this, we using this command, then enter. Okay, now this command is in running mode. Okay, this is the command. Gene ID, their chromosome, starting point, end point. Okay, now all is thing okay. Data is also observed here. Okay, this is the complete data. 11 5901. There's total 7 column. We extract the count matrix. For extracting the count matrix, we use this command. Okay, we create a sample for the metadata. Use this command, that is the, and enter. Then next step, we build the DE sequence data set. For this, we using this command, then enter. Okay, then we normalize this file. For this, we use this command, then enter. Okay, now this normalized file is created and is downloaded in our folder. Okay, and I will show you this file. This is the file. Okay, now normalization file is created. Okay, now we check the top 10 gene expression label. For this, we use this command, then enter. Okay, now these are the top 10. For next is for variance instabilization, and for the visualization, we use this command, top gene expression level. Click this one. Now these are the top genes. We already discussed this. Now we create a histogram of this top 20 genes. We use this command, then enter. Now this is the histogram. Okay, frequency and normalization. Using this command, we focus. If you want to focus any specific gene, then you use this command, gene name, ND6, and normalization count, and this one, and enter. Okay, 1 and 5th position. To download top 100. So this top 100 gene of interest is in download in your system. I will show you. These are the top 100 gene samples. This is okay. This is the top. Now we revise what we do. Okay, first we install the package, and these are the package. First package is your biomanager and DE sequence. Okay, this head map is for creating a head map, and ggplot if you want to create a ggplot. Okay, but we understood how we use this DD sequence for the differential gene expression. Okay, for this, first you install this package. After installing this package, you load the library. For loading this library, you use this command, that is the library DE sequence 2. Okay, then this library is automatically load. Okay, then you upload the feature count data on RStudio. For this, you use this command. Okay, then you find this result. Okay, then you find this result. Okay, then you create a matrix. For creating a matrix, you use this command matrix, then count matrix greater than minus as matrix column uh, under bracket comma -1, then gene id, sample name, etc. Then you create the metadata or call data. For call data, you use this command. Okay, then you run your software. For running the software, you simply write this command. Okay, if this warning is showing, then ignore this. Okay, then create a normalization file. Then create a normalization file, you simply write this. Okay, then this normalization file is installed in your system. Okay, this is the normalization file. Okay, okay, this is my normalization file. This is the file. Okay, sample and their data. 2310 number of genes are present. Okay, now we want to see the top 10 sequences or top 20 sequences. Then you use this command top underscore gene greater than minus short norm dash count 1 decreasing equal to 0 head top 20 top underscore gene comma 20. Now these are the top 20 genes. Okay, these are the top 20 genes. Okay, now if you want to interested in any particular one gene, then you simply use this command. Okay, if you want to shorten the gene list, top 5 gene list or top 10. Now if you want to find out the some top 10 some samples, then you use this command. Okay, now hmm, now if you want to download the top 100 sequences in your folder, then simply use this command. Okay, whenever you write this command, then this top 100 genes is installed in your system. This is the top 100 gene. Okay, in this way you easily perform in this way you easily perform DE sequence 2 for the differential gene expression analysis. Here we take a here our target sequences sample is only 1. Okay, if you take more than 2 sample, then it is more appropriate for you. Okay, and here you also see the all data, your call data, count met, count, DDBC, norms, top 5 sequence, your gene of interest, top genes, top 100, and here we also distribute and also create a normalization count graph. Okay, this is all about the DE sequence 2. Now we discuss the application of next generation sequencing. Okay, this is the last part of our video. Okay, first application in whole genome sequencing. Okay, how whole uh, by the help of NGS, we easily find out the whole genome of any organisms. Okay, WGS or whole genome sequencing provide a complete picture of an organism because genome is a complete set of genetic material that include the coding as well as encoding part. Okay, it helps to identify the genetic variations like single nucleotide polymorphism, insertion, deletion, and structural rearrangement. Okay, whenever we sequence the complete genome, then this is very useful for the disease causing mutation to identify the disease causing mutations, comparative genomic between the comparative genomics between the species and individual to find out the evolutionary relationship, or in cancer genomic genome sequencing to detect the mutation, translocation, or your copy number variant. Okay? These are the application of NGS in WGS. Now, next application in whole exome sequencing. Whole exome sequencing means here we target only coding region. In gene, only 1 and 2% is a coding part. Okay, where most of this coding region is responsible to causing a number of diseases. Okay? Whenever we sequence the whole exome, then this is very beneficial for diagnosing the Mendelian disorders like single gene disorder. Also helpful to discover the novel gene associated with the diseases and cost-effective alternation to WGS for the clinical genetic testing. Okay. Next is your transcriptomic. Next is your transcriptomic sequencing. Means by the help of NGS, we easily sequence the RNA. Okay. We sequencing how we sequence the RNA. First we isolate the RNA, then we convert this RNA into the cDNA, then sequence. Okay, sequencing of cRNA generate the rRNA to analyze the gene expression level or transcriptome value, and this is also very beneficial in medical sector. Like by the help of this, we easily find out the differential gene expression. That how we find out the differential gene expression we already discussed in detail. Okay, identify the alternative splicing, fusion gene, non-coding RNAs, and this is also very helpful to study the cell type, specific expression, regulatory network, and this is also very helpful for the drug designing, for the drug discovery, for the vaccine designing, or to design the biomarker, to design the disease marker. Okay, this is the application of NGS in in RNA sequencing. Now, target sequencing means by the help of NGS, we easily sequence the target gene. Let's suppose we focus only a specific part of gene. So first we isolate that particular part, then we go to the sequencing. Okay, and this plays a most important role in clinical diagnostic and pharmacogenomics, in carrier sequencing, in cancer genomics, in vaccine designing, in drug designing, in etc. Okay, like clinical designing means mutation detection. Pharmacogenomics means to understand how the gene is variant and affect your drug, and carrier screening means to find out the genetic disorder. Okay. Now, next order in metagenomics. Metagenomics means sequencing of DNA directly from the environmental sample without culturing the organisms. Okay, and this is very helpful for study the microbial diversity, discover the novel microorganisms and their metabolic pathway, tracking the antibiotic resistance gene, or human microbe analysis. Okay. Next application in epigenomics. Epigenomics means NGS based technique like Y-sulfate sequencing or CHIP sequencing are used to the DNA to study the DNA methylation and histone modification, and this is very helpful for understanding the gene regulation in epigenetic inheritance, in cancer research, etc. Now in cancer genomics it plays a most important role to to co- to to comprehensive study the tumor genomic, or to understand how the cancer is develop and what is the heterogeneity. Okay, and it is very helpful to detect the somatic mutations, copy number of variation, and gene fusion. And this is also plays a most important role in personalized medicine. Okay, now next application in clinical diagnostic. In clinical diagnostic means NGS is increasingly used in clinical and personalized medicine to identify the genetic variation related to the diseases. Okay, like prenatal and neonatal screening, inherited disease testing, pathogenic detection, and rare disease diagnosis. Okay. Now, next application in pathogen genomics and public health. NGS, by the help of NGS, we are rapidly identify and characterize the pathogen. Okay. It is very helpful to outbreak track like influenza, COVID, COVID virus. It is also very helpful for antimicrobial resistance, gene detection, vaccine and drug discovery, and real-time genomic surveillance. Okay. Now, next application in agriculture and environmental genomics to improve your crop yield, resistance, and environmental susceptibility. Okay. Next application in forensic genomics. Forensic genomics means NGS allow the identification using the trace amount of DNA and mixed sample. Okay. Next application in synthetic and biological biotechnology. NGS are very helpful to design, to validate of engineer biological system like genome editing validation, a best technique is your CRISPR. By the help of NGS, we also validate the CRISPR-Cas system. Okay, pathway engineering or protein engineering. Okay, these are the application of NGS. Okay, now we discuss about the Now we discuss about the role of NGS in microbial taxonomy. Microbial taxonomy is the science that are used to identify, to classify, or to naming the microorganisms. Okay? It is depend upon the microbial characteristics, biochemical testing, and 16S RNA sequencing. Okay? This is the base of NGS-based taxonomy. Okay, the traditional classification taxonomy method is depend upon the non-cultural microbes, high similar and noble species, and microbes with complex diversity. Okay, and this method have some limitation. Okay. Okay. However, these traditional method have some limitations, and this is modern method of microbial taxonomy is based on this microbial characteristic, biochemical test, and 16S RNA sequencing. Okay. Traditional method have some limitations. Limitation especially for non-culturable microorganisms, non-culturable microbes, high similar or noble species, and microbes with complex diversity. Okay, but by the help of microbial taxonomy that is based on a NGS, we easily classify or to find out the evolutionary relationship of non-culturable microorganisms, highly similar or novel species, and relationship between all these microbes with complex diversity very easily. Okay, this NGS revolutionized the microbial taxonomy that will provide the deep and comprehensive and cultural independent analysis of microbial diversity or evolution. Okay, now how we use NGS in microbial taxonomy. Okay? NGS enables the large scale sequencing of microbial DNA or RNA directly from the sample. Your sample may be soil, may be water, may be gut, without isolating and culturing by the help of third generation sequencing like PEG biosequencing and Oxford sequencing. Okay? These are the NGS approaches like Applicon sequencing, 16S RNA, ITS, and 18S RNA. Okay, description, target a specific marker using the using the primer, and uses taxonomy for identify the bacteria to fungus, archaeas in genus at species level. Okay, let's suppose we want to identify the bacteria, fungus and archaeas at their genetic and species level, then we use applicon sequencing. Okay. Next, whole genome sequencing. Whole genome sequencing to complete genomic and pure culture isolation. Okay. Let's suppose we define the specific boundaries, strain level identification. Okay. Now, metagenomics. In metagenomics means sequence total DNA in a sample. And this is very helpful for characterization of entire microbial composition, including the unculturable species. Meta-transcriptive means we sequence at the RNA level. Means we isolate, we sequence the complete RNA of an organism. That is very helpful for reveal the active microbial taxa and their function. Okay, because we sequence the RNA. RNA is responsible for the expression level, for the expression of your gene. Okay. Now, pangenomics analysis means compare the genomic of multiple strain or species. That is very helpful for refining the taxonomy and identify the evolutionary relationship. Okay. Now, we discuss the application of NGS in microbial taxonomy to first application to accurate species identification. Let's, by the help of 16S rRNA sequencing, it is a very high resolution identify the bacteria at their species and strain level. For example, we easily distinguish the two strain of your bacteria like E. coli and Shigella, which have a 99% similarity, but by the help 99 16S similarity, but different genomic content, and it is possible by using whole genome sequencing. Okay, and for this we use the software like QI mean to data to saliva and green gene database. Okay. Next, to discover the novel microbial strain. Okay. Many microbes in nature cannot be cultured. That is known as a microbial dark matter. But by the help of metagenomic sequencing, we easily sequence the DNA of that particular uncultured microorganisms. Okay. For example, by the help of NGS, we easily sequence the candidate CRP organisms. We easily identify the CRP organisms using the metagenomic assembled genome. Okay. For this, we use the software like CheckM, GTV-TK, PROCA, and ANI to find out the average nucleotide identity. Okay. Next is your phylogenetic analysis and evolutionary relationship. NGS also reconstruct the phylogenetic tree of hundreds and conserved sequences. Okay. For this and provide a most robust and accurate evolutionary relationship. And to construct the evolutionary tree, we use a software like Mega12, FastTree, etc. Mega12 is a very helpful software to construct the phylogenetic tree. Okay. Next is your redefining the microbial taxonomy. Whole genome sequencing has led to reclassify of microbial taxa. For this, we use ANI, that is the average nucleotide identity. If more than 50% means same species. DDH means digital DNA hybridization. Is the result of DDGS is more than 70, it means same species. AI, that is the average amino acid identity for the gene level classification. Okay. These are the tools, and this is the way by which we predict the result, if we predict the result by the help of this software. Okay. Next is microbes and environmental taxonomy. Means NGS allow identify the microbial taxine environmental sample like soil microbes, marine ecosystem, and human gut microbiota, microta, microbiota. Okay, this analysis is very helpful to understand the ecological diversity, species richness and abundance, and evolutionary of symbiotic and pathogenic relationship. Like human microbe project, like that is known as the HMP, use the 16S and metagenomic NGS to define the microbial composition in healthy and diseased individuals. Okay. Now these are the most useful bioinformatics software. Like to check the quality, we use FasterQC and FasterP and Trimometric. For assembly, we use SPDES and MegaHit. For taxonomic classification, we use this Kraken 2, MetaPHIAN or QIN Min2. For phylogenetic, Mega Software and RAXML. For gene annotation, POCA and RAST. POCA and RAST. For species boundary for metrics, we use ANI calculation or GGDC. This GGDC is a free web tool. For data analysis, we use Sliva and CBI ref sequence, GDDV, green gene और होगा सेट कि नहीं होगा बस Now we discuss about the advantages. Like to detect both culturable and non-culturable microorganisms, enable find the resolution species and strain identification, provide the complete genomic context, rapid and cost-effective method, and support the evolutionary relationship. Okay. Now we discuss some limitation and challenges. Means high computation and storage requirement, short read may causing the assembly error. Data wise means incomplete reference genome may causes the misclassification and require the bioinformatic expertise. Okay, these are the limitation of this. Okay, these are the limitation. Okay, thank you so much. Thank you so much. Thank you so much. This is all about the today's session. Thank you so much.